Just a second. I don't know why this thing is not showing on the screen. My file. Okay. Can you see my screen here?
Yes, sir, it's visible.
Sir.
Okay. And
Yes, sir.
let me see. Now full screen.
Yes, sir.
Let me get my pointer just like I always do. And let me also go to some presenter. This makes life a little easier for me. Okay. So if you recall, last time we stopped at the point where we were talking about how to detect the bacteria which has actually taken up and is having the plasmid that you have made in a recombinant fashion. Okay. In other words, a plasmid which contains the sequence of DNA which you want to amplify. Okay. So, and we said that this one method of doing this is called as replica plating, which actually follows the phenomena of what is called as insertional inactivation. Okay, now just before we go forward, we'll quickly go back. If you remember, in this method of making, trying to clone a gene, in this case, the gene I want to clone is this B gene, which is shown here in yellow with a little cross on top. Okay. I want to amplify this gene. So what I did was to cut this gene out from wherever, from whatever source using a restriction enzyme. And when I did that, I got a piece of DNA which had a very specific ends depending on which restriction enzyme I used. Then what I did is I chose PBR 322 as a vector. And what I did is, fortunately, in this case, this vector has got a site which can be cut by the same restriction enzyme which I used to take out this DNA. And this site is present in the middle of this, what is called as tetracycline resistance gene. Okay? A gene which produces a protein which allows the bacterium which carries this plasmid to show resistance to tetracycline. Now, when I do the cutting and pasting, that is, cut the vector and then mix it with this already cut out piece of DNA and then ligate it, I said that in the mixture where I have put cut vector and cut DNA, I'll get a lot of combinations which might end up getting sealed when I treat it with ligase. Okay, and two combinations which are of interest to us. Okay, are one where this gene has actually inserted itself in this area which was cut to give you a recombinant plasmid which looks like this. It has the same origin of replication. It still has an intact gene for ampicillin resistance. But here, where the gene for TC was there, I have this sequence which has got inserted. Or what I could have got is that it is cut open when I treated with restriction enzyme. But even though I mixed it with this piece never got here when I treated with ligase, and it simply resealed itself. Okay. And that original plasmid will have the same characteristics that we started with. It will show resistance to ampicillin, and even this gene is intact, which means that later on when I take a mixture of all the things which are there, when I cut this thing, cut this thing and mix them together and seal them with ligase, there will be some bacteria which might end up taking this up. There might be some bacteria which might end up taking this up. Okay? And what I might find is that those bacteria which have taken up whichever plasmid, they'll show typical characteristics. For example, if a bacteria has taken up this one or this one, it will show resistance to ampicillin because that gene is still intact. That's why, with respect to ampicillin resistance, you said yes with respect to this recombinant plasmid. We also say yes, it will be resistant to ampicillin with respect to this plasmid. But when it comes to tetracycline resistance, I'll find that this recombinant plasmid will show no resistance to tetracycline. And in other words, it will be sensitive to this antibiotic. The reason it is sensitive is that now that original protein which gave the bacteria the property of being resistant to tetracycline is now disturbed and is disturbed by insertion of this yellow thing. So now you get a non-functional protein which is unable to take care of tetracycline, and therefore the bacteria gets killed in the presence of tetracycline. Now that doesn't happen here. So here the resistance to tetracycline is still intact, just like it was in the original plasmid, which means that all those bacteria which take up this will show resistance to 2 antibiotics, and all those bacteria which take up this plasmid will show resistance to only one, which is ampicillin. Okay. Now, how do we distinguish this? What we initially do is take all the culture that we have after doing a transformation experiment. That is, remember, I need to introduce it into bacteria by either doing heat shock or electroporation or calcium phosphate. After I've done that experiment, what I do is take that entire culture that I have and I grow it in a plate which contains ampicillin. Now, what this will do is that every bacteria which has not taken up any of this plasmid, which is free of plasmid, will automatically die. Okay, because there is ampicillin. So the only bacteria which will survive in this plate are those bacteria which contain either this recombinant plasmid or they contain the original plasmid. Okay, so that is what is shown here. Okay. Okay, so when I do the initial culturing, I'll see a bunch of colonies, but these colonies are colonies of bacteria which contain the plasmid, which is either the original plasmid or which contains what is called as a recombinant plasmid. Okay. Now, in the next step, what I do is, as I say, lift this colonies. Okay, the way I lift this colony is that I take a sterile pad and I put the pad over this plate after opening the Petri plate under sterile conditions, so that a few members of each of these colonies end up sticking to this pad. And then, keeping this orientation intact, what I do is I take that pad and press it on a plate of media which contains tetracycline. Now, what will happen is, everywhere you had members of each of these clones on this pad, you'll get a little replica of those and they'll stick to this new plate. Now, the issue is this plate has tetracycline. So, what will happen is, among all those colonies here which showed resistance to ampicillin, the only one which will grow in this plate are the ones which are resistant to tetracycline, which means it will only allow growth of those plasmids or those bacteria which contain the original plasmid, because that is the one which is resistant to tetracycline. While all those bacteria which actually contain the recombinant would die off, okay, and they'll get killed. Now, luckily, what you have done is not taken the original plate and added tetracycline to it. So this plate is still intact, remember, because you have only taken a little copy of this by lifting the colonies and putting it here. So now what you do is you look at this plate and you look at your original plate. Okay, that is what we are doing here. So this is a plate where colonies are growing which are resistant to tetracycline. Okay, and this is the one which is the original plate. So what you do is you take out those colonies which are there in this plate, but which are not there in this plate, and you know that those are the ones which were resistant to ampicillin, but which are sensitive to tetracycline. And therefore, those are the three bacteria which contain recombinant plasmid. And then all you have to do is very carefully take little, samples from these three colonies, put them in fresh medium, grow them in the presence of ampicillin, okay, and then they will start growing. Okay, the only mistake you cannot make is take these things and put them in tetracycline, because if you do, they will die, and you know that, right? So, and then once this grows, then you take out the bacterium, open it up, get the plasmid, and when you cut it with the same restriction enzyme that you used in the previous step, you'll get this piece of B which will be released, and you'll get your DNA amplified. Okay? Is that clear?
Yes,
Everyone?
sir.
Yes.
Okay.
Yes.
Now remember that when we were talking about plasmids, we said there's another plasmid which is used very commonly, and that plasmid is called as the PUC18 plasmid, or sometimes it is called as PUC plasmid. Okay. Now this is very interesting. This also has ampicillin resistance, but in addition to that, instead of having tetracycline resistance, it has got a gene which produces, as I said, beta galactosidase. Okay, and this beta galactosidase is able to take lactose and break it down to galactose and glucose. So here something interesting happens when you do cut and paste, right? Now let us say I insert a gene which I want to amplify, and I insert it in this region here, okay? Because that is where all the restriction sites are. So I can cut it open, put my gene and seal it back. What will happen here is that all the plasmids which are recombinant, that means which contain the foreign gene somewhere here, will now show resistance to ampicillin, but they will not be able to produce this enzyme, okay, what is called as galactosidase. So now see how they do the selection. Now, after doing cutting and pasting, what they will do is initially grow the colonies in ampicillin. So when they grow it in ampicillin, what will happen? The bacteria which contain the original plasmid, which is this PUC18, and the bacteria which also contain the recombinant plasmid, which will look like this, but will have a little piece inserted here, will both grow. Okay? And after that, what you do is very interesting. In the same medium, which is still containing ampicillin, okay, you simply add a compound which will be metabolized by this galactoside, okay. And typically what they do is they add an analog of lactose, which when broken down by galactosidase will give a color, okay. Now see what happens when you do that. Now, see what I've done? I've taken a piece of DNA, I put it in this plasmid, and I put it where? In the size, in the place where lag Z is there. Now, what will happen is I'll get a recombinant plasmid, but the recombinant plasmid will not be able to produce, as I say, uh, galactosidase. Now, which means now that when I take all of these bacteria which contain either the original or the new, and I grow it in medium which contains both ampicillin as well as I said, a compound which can be metabolized by galactosidase. It is called as X-gal. Okay, see what will happen. All those bacteria which contain no plasmid at all, okay, they'll be sensitive to ampicillin, so they'll simply die. So there'll be no growth. Okay, let us say that bacterium falls here. Now the media has got ampicillin and X-gal, so they'll simply die because there is no ampicillin. The only ones that will grow are the ones which have the plasmid with no insert. That is the original plasmid and the plasmid with the foreign DNA inserted in the lag Z. Now see what happens to them. The plasmid, which has got no insert, which produces galactosidase, is able to break down this X-gal to a compound which is blue. And those colonies start appearing blue. So these blue colonies that you see are all bacteria which contain PUC18, which is still intact and which is producing beta galactosidase, which is breaking down this X-gal to give you a blue colored compound. Now see what happens to all those bacteria which contain recombinant plasmid. They're resistant to ampicillin, so they're able to grow, but they're unable to break down this X-gal to compound which is blue, so those colonies do not appear to be colored. So here in one experiment, that is by taking this mixture of plasmid and simply growing them in media containing ampicillin and X-gal, I am able to quickly choose which is colored, which is non-colored, and the non-colored ones are the ones which contain my DNA present in the PUC18 plasmid. So all I have to do now is pick these colorless colonies and start growing them in media containing ampicillin. They'll grow very nicely, and then later on I can take out the plasmid, cut out the DNA that I want. Okay. So the other day somebody asked me, okay, is this the only way, okay, by looking at colony characteristics here to find out whether my bacteria has got recombinant DNA? So here is another way, again, colony characteristics. And plus, as I said, the other thing you can do is actually do sequencing. Okay, is everyone clear?
So,
Yeah,
Yes, sir.
so in this
yeah, somebody.
media, we have to add even glucose now because the recombinant plasmid won't have the gene to break down galactosidase, so it needs some energy providing media, na?
Correct. You will use glucose. Okay, you're right. You will need glucose because these fellows are unable to use lactose as a source of energy. Okay. Now, yeah, very
Yes,
correct.
sir.
You will need to provide glucose. Okay. Now, see, you also have to realize that the story that I told you about how to do this cutting and pasting and making recombinant plasmid is almost like film ka story. Okay, and everything seems to be there. You have a gene which has got nice two sides on both sides, which will be cut by the restriction enzyme that you are interested in, and everything will happen correctly. In real life, that doesn't happen. Okay, many times what happens is the gene that you are interested in amplifying doesn't have on both sides a sequence which can be cut by the same restriction enzyme. Okay, plasmid here no problem because you need one side. This plasmid is made by you. Okay, so you know exactly how the plasmid is. But with respect to the gene, sometimes you might be unlucky. Okay, there is a gene, but on both sides you do not have the restricted sequence that I want. So what do you do in that case? Okay, sometimes what is done is in that case, what you do is see whether on both sides of the gene there is a sequence for some restriction enzyme which does what is called as blunted cutting. Okay, blunt end cutting, if you remember, is flush end cutting. Okay, so see, for example, I got a piece of DNA now which is cut by on one side by a restriction enzyme which recognizes a specific sequence, but it cuts in a blunt fashion. And fortunately, on the other side, it is also cut by a different restriction enzyme, maybe. But what is important is that also gives a blunt end. Now, what I do is something very interesting. I take this piece of DNA. I know my gene is in between, but I know I do not have sticky ends. And what I do is very smartly uh, take this enzyme. If you remember this enzyme called terminal transferase, which is able to add nucleotides in a template independent fashion, kind of the last enzyme we looked at
Yes, sir.
before we started. So you do take that enzyme and to it you add deoxy CTP. So see what it will do. You start putting a tail and put a tail which is CC. Correct? Now what you do is you take the vector, cut that also with a restriction enzyme which gives blunt ends. But to that, you add terminal transferase and put GTP. And when you do that, what will happen? That will add GGGGG to the 3' ends. Now the beauty is, this now is exactly complementary to this, correct? So when I put these two things together, this G and C will come here, and this will like circularize, and it will bond with this G, and I'll get a nice plasmid which is now recombinant. Okay, and now whatever this remaining thing, this little gap is usually corrected by adding, if you want, another. When you seal it, no, what will happen very interestingly, during the sealing, a little repair activity also occurs sometimes, and this G and C will all get sealed correctly, and you'll get a plasma. Okay, now this is referred to as homopolymer tailing. So essentially, what you are doing is homopolymer tailing means same basis you are putting as a tail on, but you put it complementary on the vector and the insert. Okay, is that clear? Or sometimes what people will do is even more interesting. They will take a blunt piece of DNA which you have got, which actually contains a gene, and to that you actually ligate a specific sequence that you're interested in, but which is also blunt, and put it together. Okay, remember ligases will also do blunt end sealing. So when a ligase cuts and puts these two together, what you'll find is at the end of this sequence, you'll have a very specific sequence, but that has a palindromic sequence. So now you can cut it with a restriction enzyme that you're interested in. Okay. So you actually what you do is manufacture ends that you want so that you can use the restriction enzyme of your choice, which is matching also with the vector later on. Okay. So there are a lot of tricks people will do in case my gene is not flanked by two sequences of restriction enzyme. Okay. Is that clear? Okay. So there are little tricks you have done. So see what you have done here. I do not have a sticky end, so I do a blunt end ligation. But look at this piece here. This has GGA-TCC, which is actually palindromic, which can be cut by BAM-H1. So when I join these two together, now I have a piece which I can get a sticky end out. Okay, everyone.
Yes, sir.
Okay.
Yes, sir.
So, so what we have looked till now is how you can take a piece of DNA, very systematically put it into a vector, which is going to carry that piece of DNA, take that DNA, introduce it into a bacterium, select the bacterium which has taken up the DNA because of unique properties that your vector has, and then amplify that bacterium which is containing that piece of DNA, making millions of copies when it grows, and then later on open up the bacterium to isolate the plasmid, cut the plasmid with a restriction enzyme, and get back that piece, and now you'll have millions of copies so that you can do whatever studies you want to do. Okay, so in short, essentially what we looked at is that DNA molecules can be digested with enzymes called restriction enzymes, which actually renders them manageable for what you call as cloning. Then what you do is the products of this digestion, as I call it, are inserted into a vector, where the vector is also cut by the same restriction enzymes. Then you seal it with ligase and then introduce the recombinant DNA into a host cell. Okay, and when you do this transformation, then each cell which gets this rDNA is technically a clone. And what you might do then is take these clones and grow them so that thousands of copies are made as DNA starts to replicate, and the DNA is given to the daughter cells. And finally, what you do is DNA, extract it out, purify, cut out that foreign DNA that you want, get it in large amounts, and then you can do whatever investigation that you want about the sequence, the structure, function, etc., etc. Okay. Now, Such a elegant technology, okay, and maybe some of you have been also told about it in your 11th and 12th, okay. So many of you are not totally unfamiliar with it. Many of you know about it. But the irony is, as of today, we rarely use this method to amplify the DNA. Okay, and the simple reason uh, for not using this method is one that it takes too long. Okay, remember, I have to first grow the bacterium in order to get the plasmid vector. Okay, uh, then I have to take that plasmid vector and I have to cut it with restriction enzyme. I also have to get source DNA from somewhere, cut it with restriction enzyme, then put it both together, treat it with ligase, then I do the transformation experiment. Okay, and then after doing the transformation experiment, I have to wait for 1 or 2 days for the colonies to start developing. Then, if I'm doing replica plating, I have to take those colonies, put it in a fresh plate, wait for those colonies to develop. Or if I'm doing what is called as blue-white colony, after the first plating itself, I might get answers. But whatever be the case, after I've got the colonies, I have to again grow them. Okay. Again, when I grow them, it takes time for it to multiply. So typically, this entire experiment of what is called as cloning a DNA or amplifying a DNA will take at least a week to 10 days. Okay. From the point where you have started, with a bacteria, making it grow to get the original plasmid, then cutting, pasting, and everything. And that is presuming everything goes right. Okay, you get no contamination, and actually bacteria take up the recombinants. Sometimes these are all probability events. Okay, so and therefore it's a tedious job. You have to keep track all the colonies and everything, and it takes anywhere from a week to 10 days to get to that point. Okay. As of today, there are techniques which are much faster than that. Okay, and because they are much faster than that, and also as fairly reliable, what people do now is rarely use this classic what do you call it, cast, paste, transform, grow, select the colonies, etc., as a routine method. Okay, you can still use it in very specific cases where this other method may not work, but for all practical purposes, this original method that I've talked to you is used very infrequently. What we end up using nowadays is a method which is called as PCR, or polymerase chain reaction. Okay. Uh, uh, Anybody has heard of this technology before coming to this class?
Yes, sir.
Polymerase chain reaction.
Yes, sir.
Yes, sir.
Yes, sir.
And do you know what it actually
Yes, sir.
does? And a little bit about it. Anyone want to
Yes,
talk?
sir.
Unmute
Yes,
and
sir.
Sir,
say
it's
something.
used to amplify DNA sequence.
Like first, like we
Uh,
have our DNA, so
uh,
we heat it at a temperature of about 94 degrees Celsius, so that the all the hydrogen bonds are broken, and
mm-hmm,
then we add the primers and the other required enzymes, so that
mm,
the can say a copy of it is being made, and then we
mm-hmm,
again heat it to a temperature of 72 degrees Celsius, where we add the tag polymerase enzyme.
mm-hmm. And what is the stack polymerase enzyme?
I don't know the use of it, but it's like it
Mm,
is required.
it is required. Okay. Anyone else has heard of this technology? Want to say something about it?
So Taq polymerase is
I
used in the
think
process of annealing.
It is in the process of annealing. Okay. Let us see. Huh? Okay.
it is extracted
Yeah.
from thermophilus at this.
Uh, okay, it is extracted from thermophilus something. Okay, but I'm happy a few people have at least heard of it. It was originally called polymerase chain reaction, uh, and it became so famous and so useful that nowadays people actually sometimes call polymerase chain reaction as people's choice reaction. Okay, indicating the popularity of this method. Okay, and it is used to amplify a very selected region of DNA from very little starting material. Okay, in fact, even if you have one double-stranded copy of a piece of DNA in which there is a sequence that you want to amplify, you can do so. Okay, in fact, it is used in forensics. So that's why they say, no, nowadays, even if you leave one cell at the crime site or a piece of hair at a crime site, you can actually get to know who was there. Okay, because you can amplify the DNA by this method called as polymerase chain reaction. Okay. Now, this method has got some interesting history to it, okay, and nice stories around it, okay. And one of the stories has an Indian connection to it also, okay. Many of you must have heard of a scientist called Hargobind Khorana. Anybody has heard
Yes,
of
sir.
this?
Yes,
Yes,
sir.
Hargobind
sir.
Khorana. And what contest? Yeah, in what contest have you heard him?
So he won the Nobel Prize.
Yeah, he won the Nobel Prize for discovering uh, which
He discovered
codon.
codons.
Correct. Which codon codes for which amino acid. Okay. He not only discovered what codons are, but we actually did research to actually tell you. Remember, AUG is coding for methionine, and so on and so forth. Okay, very brilliant scientist. Okay. Now look at. I'm going to very simply tell you what PCR is before we go into details. Okay. What is done in polymerase chain reaction is you take the original piece of DNA which contains the sequence you want to amplify. You see the sequence which is shown in blue? That is a sequence I want to make multiple copies of. Okay. So what I do is I first heat the DNA to close to 80 to 90 degrees. Okay, in reality, you might do even up to 95. So when that happens, these two strands will break. Remember that we talked about how we melt DNA by heating it, and when you heat, the hydrogen bonds will break. So these two strands will now be separated like this. Okay, all the hydrogen bonds are broken. Then what you do is to this hot mixture, you add two different primers, but these primers are designed in a very interesting fashion. Okay, look at this yellow primer. It binds to this region. Okay, so its orientation is 5 prime to 3 prime in this direction, because remember this strand is 5 prime to 3 prime in this direction. Okay, so this primer is 5 prime to 3 prime in this direction. In the same manner, you choose another primer which is complementary to the sequence somewhere here and which binds to this region, but that is also designed in such a fashion that its 5 prime end is here and the 3 prime end is facing towards this primer. So the two primers actually face each other in a sense. Iska OH group and Iska OH group are looking at each other. Okay, so the directionality is 5 prime to 3 prime like this and 5 prime to 3 prime like this. And once you do that, okay, adding this primer, you reduce the temperature of the mixture. When you start reducing the temperature of the mixture, what you might get is sometimes these two DNA pieces will come together. Okay, so you'll get back your original double-stranded DNA. Sometimes you'll get one piece of DNA which is like this, uh, and sometimes you'll get only one piece of DNA which is like this. But you'll also might get a mixture where you have double-stranded DNA, where this section here is interrupted by these two primers. Okay, remember these are all complementary here. Okay, but because this primer is bound here, this cannot come here because of this little gap there is there. Now, what you do is you add DNA polymerase. When you add DNA polymerase, because it has got a template and it has got also a primer, it starts to multiply. But what it will do now is add bases from here till it reaches the end. And to this one, it will add bases from here till it reaches the end. So now, what I will get as a partner to this strand is a shorter strand, but which is still complementary to the sequence that I'm interested in. Same happens here the strand I have just made is shorter than the original because it started from here, but then it contains that sequence which I am interested in. Okay. Now this experiment to show that you can do it like this was actually done by Hargobind Khorana. Okay. And he said that, I can take DNA, I can separate it by heating. Then if I'm very intelligent, I can add 2 primers. I design it correctly so that it brackets the sequence that I want to amplify. Then I add polymerase. Then I'll get copies. Okay? And then he also proposed that if I keep repeating this experiment, I'll be able to get more and more copies of this sequence that I'm interested in. Okay, which means what? Now I will take this, I will do the same experiment. So now this will melt with this will melt with this, then I'll put the primers, and then again the same thing will happen. Okay, and you'll find that the sequence will be again and again replicated. Except he was unable to actually do it and show it unequally. So he theorized this, but he was unable to actually physically do and show. And the reason for that is very simple. At that time when he proposed the idea, DNA polymerase was very costly. Okay. Now think of this. I heat DNA, I add the primer, then I cool it so that it can join to this DNA. But then when I want to add DNA polymerase, I have to bring the temperature down to close to 35-40 degrees, because that is where this DNA polymerase works. And when I do that, I'll get this polymer. Now, if I want to repeat the next cycle, I have to heat again to 95, right? When I heat to 95, this polymerase gets inactivated because it's a protein, it denatures. So if I want to repeat this for another round, and I heat, then I again put primer, bring the temperature down, I'll find that I will not be able to do polymerization unless I add another dose of polymerase. Now this polymerase was extremely costly when Khorana proposed this idea. So even though he wrote about it in a paper, he never actually proved, okay, you could keep doing this. And it kind of stayed that way. And people thought, okay, yeah, that's a way to amplify DNA, but it is so costly because DNA polymerase is so costly that we will keep on doing that original, put in bacteria, put in media containing ampicillin. I cannot afford the cost of this polymerase. So this technology, even though proposed, never actually got anywhere. Okay. Now, a few years later, okay, sometime in around the 80s, early 80s to middle 80s, as they say, another scientist was also having this issue of trying to amplify DNA, called KRI. Apparently, they say that he got this idea while driving home one evening from work. And he was driving across the hills of California. Okay, in this valleys, they have this up and down roads which are very, very beautiful. And he got a brainwave at that point. Okay, and he said that I can amplify this DNA by doing this repeated rounds of melting, annealing, and adding polymerase. But he had the knowledge of a polymerase which was stable to high temperatures. Okay, and this was a polymerase which was isolated from a bacteria which actually grows in boiling water, a bacteria by the name Thermus aquaticus. Okay, and therefore the polymerase which was isolated from this enzyme, because this enzyme also needs to replicate, right? But it is growing in hot water. So every protein that this bacteria has does not denature at 90-95 degrees, because if it did, the bacteria would have been dead by now. So every protein this bacteria has evolved to be functional at very, very high temperatures. So this KRI simply said that if I take this tag polymerase, okay, and why I see the name tag polymerase from Thermus aquaticus, then I should be able to do this repeatedly without the need to add polymerase again and again. Which means now this method will require only 1 aliquot of polymerase, because this polymerase that I have is stable to heat. And that was the basis for him proposing this polymerase chain reaction. Okay, the reason it is called chain reaction is because if I start with one copy which has got this two strands which are complementary, when I do one round of this, I get 2. When I take this and I take one round of replication, I'll get 4. Then 4 will become 8, 8 will become 16, 16 will become 32. So basically, I'm going to get this what you call as amplification, which is going to be increasing proportionately as if it's a chain reaction, and therefore the name polymerase chain reaction. And in fact, he was awarded the Nobel Prize for this in 1993 for what is called as pioneering work. Okay, and but ironically, there were a lot of people who are on the side of Hargobind Khorana, who said that he had also proposed this much before KRI, and therefore there was a big court case between KRI and the company which KRI represented, and another company which said, okay, no, Khorana should also be getting credit for this. Okay, but unfortunately, Khorana himself, who was alive at that time, didn't want to take part in these proceedings, because he said, for me, it doesn't matter. Science is more important. Whether I did it or not, and whether I should get the Nobel Prize, I don't care. I already have a Nobel Prize. Okay, so he never got into this litigation and thing. But it's an interesting story that we credit KRI with this, but somewhere in the background, Hargobind Khorana and his group had also proposed very similar methodology. Okay, so now PCR is used in mol bio to amplify small sections of DNA. And what is amplifying small sections of DNA also called? Cloning, right? So essentially, it is going to do exactly what you intended to do using those plasmids and vectors and stuff like that. And you can now make millions of copies in starting from a very small amount of DNA. Okay, so now more specifically, what you do is you take double-stranded DNA. You first do denaturation. So the first step is called as denaturation step, and that is done by heating to 94 to 95 degrees centigrade. Then what you do is you use very, very specific primers, and the primers that you use are actually designed to bracket, as they say, or to capture the length of sequence you want to amplify. So let us say I want to amplify this length here, then one primer will sit here in the 5' to 3' direction. The other primer will sit here in 5' to 3', but face each other. Okay, now remember, this is very, very important. If I design the primer wrong and it is facing the other way, then what will happen? You will not get amplification of this, but this primer will end up sitting here, and this primer will end up sitting here, and it will go like this, and this will go like that, and the piece you want to amplify will just remain like that. So make sure the two primers are actually facing each other. But now they have to now bind to this complementary sequence here, and you do so by cooling it to 50 to 56 degrees. Okay? And once the primers are annealed, you add your Taq polymerase and heat it to 72, which is optimum for the Taq polymerase. Okay, so unlike our enzymes, which work at an optimum of 30 to 35 to 37, this Taq polymerase works fastest at 72. So you heat it up a little bit, and boom, it'll add the complementary strand, and it'll add the complement. Then all you have to do is take these two pieces and run it through the same cycle again and again. But the good thing is I don't have to add enzyme again and again, because Taq polymerase is temperature resistant. Yeah.
So in the second cycle when we are doing, then in that case the primer which will be added will be different, right, from the first generation?
No, exactly the same primers will be added. Okay, I'll show you what happens. Okay, it might appear that you need to add different primers, but you actually don't. Okay, I'll show you that. Okay, there's a slide I have. I will tell you the next cycle how it will be. Okay, so just wait for a couple of minutes. Okay, so going further, the same thing I've shown you in terms of a table. Okay, so denaturation 94, 30 seconds, annealing 50 to sometimes 60, 30 seconds, then elongation 72, 1 minute to 3 minutes, and you run this for 30 to 35 cycles. Okay, and when you run it to 30 to 35 cycles, starting from one double-stranded piece, you can get upwards of 10 million to 20 million copies. Okay, now think how much time each cycle takes. This is 30 seconds, 30 seconds, 1 minute, and let us presume that you are going to run this for 3 minutes. So 1 minute plus 3 minutes is 4 minutes. And let us say I can do this automatically now, which is what is done. You put this thing in the instrument and the instrument will do this cycling for you. They are called as thermocyclers. Okay, and each cycle now is approximately only 6 or 7 minutes. So if I want to do 30 cycles, okay, and I presume each cycle even takes 10 minutes, okay, 10 into 30 is 300 minutes, which is like 5 hours. So I start in the morning at 10 o'clock. By evening, 5 o'clock, I will have tens and millions of copies. So imagine how much time I have saved. Okay. Now in real life, what you do is, there are little technical issues, your final elongation and stuff like that. Okay. But this is not for you. For you, the main concept is this. Okay. Now, going further, see what happens here. Okay. As I said, I take this piece, I separate it, I make a copy. Then I take there, so that will be those two pieces, each will become a copy. These two pieces, each will become a copy, and so on and so forth. Okay. From 1, I get 2. From 2, I get 4. From 4, I get 8, and so on and so forth as each cycle goes along. Okay. Now, what is more interesting is this part. Okay. Now, remember the question which was asked, do I need different primers? Now, look at this long piece of DNA. Okay. Now, important part is I want to amplify this section here. Okay. So, see what I've done. I've made a primer which is binding to this region, 5' to 3'. Then I have another primer which is binding to this region, 5' to 3'. I call this primer 1. I call this primer 2. Now, when I anneal and I add my polymerase, see what happens? This will now add DNA all the way up to here. Okay, that is shown here. And what this will do is add DNA all the way up to here, and that is shown here. Okay, so look at this piece now. Okay, so as I said, original was longer, the newer pieces are shorter because they started from here. Okay, now I'm going to take this to the next cycle. Okay, so pay attention, keep an eye on this. Top black line is big. Second is a blue line, which is shorter as you start from left to right. Then you have a shorter one, which you start from right to left. Then you have again the original black. Okay, so now look at the second round. So I start with this, right? I denature. I'll get this piece. I'll get this piece. That is from this and this. Then the third piece is this one. And the fourth piece is this one. Now see, I've added the same two primers. See, primer 1 is annealing here to the new strand. It used to anneal here to the original strand. Primer 2 used to anneal here to the original strand, but now it is annealing here to this new strand which was made from this cycle. Now when I add the nucleotides and do the tag polymerase activity, this will give you this strand which is exactly the same as this. But see what happens to this one, that will give you a shorter strand now. Okay, and what happens to this one, that will also give you a shorter strand. And again this one, I'll get this big strand. Okay, so now I have an amplified product. Okay, now if I were to keep on doing this, you know what you will find? Something very interesting happens. This small strand, which is actually the strand of interest to me, starts to increase in proportion relative to these other strands. Okay, and the only way you'll understand this is if you actually physically do this stepwise. Okay, I'm going to try and show it to you here with this blue. Okay, this is the original first primer. It went like that. This went like that. Then I took this. I took the second cycle. This again separated. This strand separated. I got this. This original separated. I got this. But in the middle, when this strand was used, I got this one, the red to this. And when this strand separated, I got this one, yellow to this. Now when I take it, next cycle, see what happens, third generation. See this, see what is happening to these pieces. The smaller pieces are getting to be more in number. So if I do 2 or 3 more cycles, no, what you'll find is that these big pieces will become less and less in proportion, and then the piece of interest to me, see this, very beautifully, that will start to increase in number, and in the end, after 30-40 cycles, this will be the majority, and this will only be 1 each. Okay, and this will be 2 each, and so on and so forth. So they don't increase very nicely, but these increase what they call, they double every time after a while. Okay. Now, although initially PCR was discovered using Taq polymerase, nowadays what you find is you can use many other polymerases also. Okay, people have discovered bacteria other than Taq polymerase which also grow in hot springs, and people have looked at their polymerases, and some of those polymerases seem to have even better functional profiles than Taq polymerase. So nowadays people use what is called as a deep vent polymerase obtained from Pyrococcus, or PFU polymerase, Pyrococcus furiosus, and Thermus thermophilus, TTH polymerase, and so on and so forth. Okay, and some of these are good because they also have what are called as good proofreading functions. So you get high fidelity, and look at some of them; they're even more stable at 95 degrees than an original polymerase. Okay, so some newer polymerases are now being used. Okay, and again, to close our discussion, when you do PCR, see what happens if you start with, say, two. When I say two, I mean the two strands which are anti-parallel to each other. One cycle, I'll get 4 strands. 2 cycles, I'll get 8, then 16, and after 19, I get 1 million. Okay, so imagine now 30 cycles. 1 million will become 2 million, 2 million will become 4 million, 4 will become 8, 8 will become 16, 16 will become 32. Essentially, the number of copies of DNA will start just rise exponentially after a while. Okay, and if I were to take number of copies and plot it with the number of cycles as a log fashion, so log of all these numbers, I'll get a nice straight line. Okay. Uh, and this is the power of polymerase chain reaction. Okay, and that's why it is also used in forensic medicine in order to just a second, I'm getting a phone call. 1 minute. Hello, hello. Who? Yeah. 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 No, 2:30 is fine. 2:30 is fine. Yeah. No, 2:30 is okay. Yeah. Okay. Yeah. Yeah. Thank you. Hello. Sorry for that interruption. I had a phone call. So is that clear, everyone? What the power of polymerase chain reaction and the little story behind it, and how it is got a little Indian connection, our own Hargopin Khorana. And what we'll do is we'll stop here for the day. I'll wait for some questions, and when we meet next time, we'll look at other technologies which are actually related to just what we are doing. That is understanding, cutting, pasting, using restriction enzymes, and so on and so forth. Okay. So any questions? So I'm going to stop sharing and see whether there is anything there, as I say, in the chat box. Okay, just let me see. Nothing in the chat box. Okay, nothing in the chat box. Anyone has any questions?
No, sir.
Here, no, I have one. Can you explain the process of making the gene of interest? I mean, we are just adding one primer at different end of DNA, and then polymerase will just go on adding nucleotides. So how can you control the length of the sequence? Okay, very nice question. Okay, now let me go back to my sharing for a second. See how you control the length of the sequence. Okay, so we do the screen share. Go back. Okay. Can you see my screen? Huh?
Yes, sir.
Yeah. Okay. So if you were to just take my pointer here. Okay. So if you think carefully, see how I control the length. The way I control the length is by actually choosing where these two primers are. Okay. Now, if I pick a primer here, let us say I pick a primer here and I pick the second primer here, what I will find is as I start amplification, the piece I will start generating will be bigger. Okay. So the way I choose what length I want to amplify and I actually control that length and not let it go further as amplification proceeds is by choosing the primers correctly. Yeah, I understand. In the first cycle, when I break this, this will go all the way to the end. Okay, wherever this is ending. But that is happening only once in every cycle. But see what happens to the pieces that you have generated from that. Those pieces, that is this piece, now that will only get extended to this point. See this? Because I use this primer to go all the way here, when this piece goes into the next cycle and the new primer binds here, then that gets extended only up to here. Similarly, this one, which in the first round went all the way up to here, when it actually gets amplified in the next run, it will get amplified only up to this point. So essentially, I control the length of what is getting amplified by choosing where these two primers are. All I have to make sure that these primers encompass the sequence I'm interested in. That is, they are not too short that my gene is incomplete or my sequence that I want to amplify is incomplete. I have to make sure it is still the sequence that I want. But once I do that, I simply have to put the two primer on the other side, and as this gets amplified, okay, in fact, I used to give this assignment to the previous classes when we used to not do online. I should tell them, do this PCR for 7 steps. Okay. See, this is one step. Going from here to here is one step. Then taking this to the next one is second. So as they say, this is first step. You'll go from first generation to second generation. In the second step, we'll go from second generation to third generation. Okay. If I do that 7 times, you'll find something very interesting. You will only realize that in the end, you get much higher proportion of this short piece of DNA. Now, if some of you want to do that on your own, you are free to do it. Okay, but I'm not going to bind you to it. But you can do it, and if you wish, you can send it to me, and I can, if you send me the photograph, what I will do is basically see if I can make a slide out of it and share it with the class, so that you realize that the easiest way to control the length of sequence which is getting amplified is to choose the primers correctly. Okay, Aditi, does that answer your question?
Yes, sir, it does. Thank you, sir.
Okay. Any other question anyone has?
Excuse me, sir. I had a question with the
Yeah,
plasmids.
yeah, yeah,
Can I?
yeah,
So,
yeah.
when we insert a gene in the vector, the length of the plasmid increases due to the extra gene, and
Correct.
instead of one palindromic sequence, we have two at the ends.
Correct.
First, we use that to ligate it. So,
Correct.
won't the cell kind of recognize that the length has increased and there's some issue with the transcription? So, like the
Okay, okay.
edges.
Yeah, yeah. So remember when we started, we said that one of the properties we need from the plasmid is that one, it should be able to replicate in the cell, correct? The other property, if you remember, was that uh, a property that inserting a piece of DNA should not affect the process of replication. Okay, and that is very, very important. Okay, and you are right that sometimes when I add a piece of DNA and the length becomes bigger, I might get a plasmid which is recombinant, but whose replication is now affected because of what I've done. Okay, so that is a concern. Okay, and I don't know whether you realize sometimes what can happen, and you can try this at home. Instead of one piece of recombinant plasmid getting inserted in the vector, I might have 4 or 5 pieces end to end which are also getting inserted, because if you remember, the sticky ends are the same. Now when that happens, the plasmid might become too big, and when that happens, that plasmid will not survive. So basically what will happen is, even though you make that recombinant, later on you'll find that that plasmid doesn't do its job, so you don't get those properties that you are using for selection. So the idea is, when the plasmid is working correctly, that it is showing, say, resistant to ampicillin, but it is sensitive to tetracycline, that means you know that you have made the correct plasmid. But yeah, what you said is a worry. Okay, that sometimes if you get too enthusiastic and try to put a big piece of DNA in that and make the plasmid much bigger than what it can handle, then the plasmid becomes non-functional. Okay, it doesn't do its job. It is not maintained by the bacteria. It throws it out or something like that. Okay, that can happen. Okay.
Yes. Okay. And sir, if a bacterial cell contains two plasmids during transformation, like we can't control how much plasmids can a bacteria take. So what
Correct.
will happen if it contains more than one plasmid?
Nothing. Actually, there are a lot of plasmids which are present in bacteria in what are called as copy numbers, which are more than one. Okay. In fact, if you look at in real life, no, there can be bacteria which contain not even one plasmid, but 4 or 5 different types of plasmids. And some plasmids are present even at high as 100 copies per bacteria, while some are present only as one copy. Okay, so those are issues which generally don't make an issue. Of bacteria, if it has more than one copy, it will be fine. Okay, and sometimes that can create a problem. Okay, for example, if a bacteria is having two types of plasmid, one the original one and other recombinant, then it will show properties that it will be resistant to ampicillin, but it will also be resistant to tetracycline. Correct? So you will actually miss that bacteria which is containing the recombinant because your original is still present in that. So those things can happen. Okay, but having more copies than one of the same plasmid is not an issue. Okay, yeah, some bacteria are known to have multiple copies. Okay,
Okay. Thank you, sir.
any other thing? Any other question? Okay, so if not, what I'm going to do is first stop my recording. Okay.